Alright, welcome to this video on Punnett Squares. Punnett Squares, all they are is just a way to predict genotype and phenotype ratios. Some vocabulary. First we have gene and allele, which you heard back in the Mendel video. But a gene is the actual hereditary information that's in your DNA, and an allele is the variations of a gene. And remember, you write a gene with an allele is written with a capital letter, and a recessive is written with a lowercase letter. So an example of a gene, the gene for hair color. And then you have different alleles of that hair color. So you have like jet black, you have blonde, you have auburn brown. These are just different alleles of that hair color. So that applies also to phenotype and genotype. Phenotype is what you physically see, so we physically see hair color, right? And then genotype is the genetic makeup. So that is this up here. What gene and what alleles make up to make this genotype. So that's the genetic makeup. That's where you get to the DNA. You physically can't see that. Genotype can be broken down into two ways, homozygous and heterozygous. So a homozygous individual, you can have two homozygous because they always come in pairs. So you can have two uppercase letters, which are two dominant, or you can have two recessive. Heterozygous would be different alleles. So this would be a dominant with a recessive. Now with this genotype, a homozygous and a homozygous dominant and a heterozygous would express the same phenotype under Mendel's principles. So take in this case if it was brown. If a dominant allele represents brown, so homozygous dominant would be brown hair color, dark brown, and then also the heterozygous of that would also be dark brown. That goes to the principle of dominance. So Punnett Squares is, like I said and mentioned in the beginning, it's just a way to predict genotype and phenotype ratios. One Punnett Square we'll be learning today is monohybrid. So in this case, we're looking at seed texture here. And another one that's also a monohybrid is a test cross. A test cross is just to determine an unknown genotype. So in this case, this would be our unknown genotype. We don't know if this flower is homozygous dominant, as in here, or homozygous recessive, as in here. So if we cross it with a known genotype, as in our homozygous recessive, then we can determine whether it is homozygous dominant or heterozygous. And that's a test cross. This is a key that I use whether I write it out or I just mentally go over it, but this is how I solve all Punnett Squares. So I have two practice problems that we're going to try. We're specifically going to be looking at monohybrid Punnett Squares, so actual human examples such as tongue rolling, earlobes, hitchhiker's thumb, facial dimples, and widow's peak. And there are some other examples, but these are the most common type examples when you talk about monohybrids. When you get into eye color, skin color, those are not monohybrid Punnett Squares. Those are a lot more difficult type Punnett Squares. So here we have a problem. In tomatoes, red fruit is dominant over yellow fruit. Design a cross between a homozygous dominant plant and a heterozygous plant. So step one is to name the gene. So we know the gene is fruit color, and that can just be determined from this first sentence here. So fruit color, and then we're just talking about red fruit and yellow fruit. So that's the gene. Then we have to determine which allele is dominant, which allele is recessive. So in this case, we have red fruit and yellow fruit. Red fruit is the dominant. Yellow fruit is the recessive. So we would write red fruit with a capital R and yellow fruit with a lowercase r. After that, we design our parent cross. So that is going to be from the second sentence. And we can see we have a cross between a homozygous dominant and a heterozygous plant. So let's design our parent cross. Starting with the homozygous dominant, it's going to be written with 2 capital R's. And then our heterozygous, I'm going to do this in a different color just to show you, is going to be with a dominant allele and a recessive allele. And that's our parent cross. Once we've done that, we do the Punnett square, and then we're able to figure out the ratios. So when you do a Punnett square, the males usually go on top, and then the female is down the side. And it's usually the same way that you write it over here on the parent cross. This would be male, and then this would be female. But as long as you're consistent throughout, it doesn't matter. So I'm going to distribute these alleles. So we'll start with the first allele pair, which is the homozygous dominant. And then we'll do the second allele pair. And then I'm just going to fill in the corresponding boxes. I'm going to distribute it. So I'm going to go ahead and distribute the top. So I distribute the top R's, and now I'm going to distribute the side. You don't have to do it in different color. I'm just showing you in different color so that you have an idea of what this looks like, how I'm distributing. So I'm just distributing everything within the box. So if we look carefully, we can see that all the phenotype for this whole F1 generation is going to be red fruit. So this whole F1 generation is going to be red fruit. So if we count it up, 1, 2, 3, 4. So we're going to have a 4 to 0, or you can just leave it 4, which is fine. Okay. Now the genotype, there are 4 probable ways of genotypes. So let's go through those. You can have homozygous dominant. You can have heterozygous or a homozygous recessive. There's always 3 ways to write it. So if we look closely, we have one homozygous dominant here, the second one here, so we're going to do 2, and then we have 2 heterozygous, one here, 2. So far it's 2 to 2, and then we have 0. So we can write that out 2 to 2. We can leave off the 0. So we have 4 for the phenotype and 2 to 2. Alright, let's try a test cross next. So this is a test cross. In cattle, black coat color is dominant to white coat color. A farmer has a black male of undetermined genotype. How can the farmer determine the genotype of the male? So this is a test cross. We do not know if it's homozygous dominant or homozygous recessive. So the gene is coat color. We have a black coat, and then we have a white coat. We know that black is dominant, and white must be the recessive from this problem. So I'm just going to use a capital B, and then a lowercase b for this, for the white coat. So the parent cross, we do not know. So the parent cross could be homozygous dominant or heterozygous. We don't know. So this is the way how you would actually determine it with what's called a test cross. We do know that white color is going to be the homozygous recessive, because the only way a recessive will ever be shown is if it's paired with another recessive, as in this case here. So we're going to perform two Punnett squares. So I'm going to take capital B, and then I'm going to take the heterozygous over here. So here is this one for the first Punnett square, and then this one will be the second Punnett square. And then let's go ahead and do the side. So we're doing a known genotype here. So this is our known genotype. So we know this genotype. Right? And then if we just fill everything in, so I'm going to start with everything in blue. Okay. And then I'll fill in the sides. So let's look at the first Punnett square. So let's figure out the phenotype. If we look at the phenotype for the first Punnett square, they are all black. So we have 4 black to 0 white. The genotype, remember you could possibly have 3, but all of them are 1. So the genotype will be 0 to 4 to 0, which you can just write out 4, which are all these. You don't have the homozygous dominant. You don't have the homozygous dominant, nor do you have the homozygous recessive. Okay? As in you just only have 4 black here. So let's look at the second Punnett square. We have 2 black, so 1, 2, to 2 white. So that would be a 2 to 2 ratio. Okay. Then the genotype would be written 0, because we have 0 homozygous dominant. We have 2 heterozygous, which is here, 1, 2. And then we have 2 homozygous recessive, which are here, 1, 2. And again, you can write that out being 2 to 2. You can just reduce that. So again, a test cross is only used to determine if you do not know the coat color. So you cross it with a known coat color. In this case, the homozygous recessive. And if that black coat cattle comes out either one of these, you can determine whether it's homozygous dominant or homozygous recessive. So here are some problems. Why don't you pause here, work through these problems. We'll look at them in class to see if you get them done. Other videos for Punnett squares will work on dihybrid crosses, we'll work on incomplete dominance, codominance, multiple alleles, and sex length. So please check back for these video sources to help you work out these Punnett squares. Thank you. Hope this helped. Have a great day.